Welcome, everyone. We'll get started about 5 after the hour. If you want to go grab something to drink or eat quick, you've got 3, 4, 5 minutes. For those that have just joined, we'll get started about 5 after the hour. So if you want to grab something to drink or eat, you've got a couple minutes yet to do that. All right, everybody, let's get started. Today's webinar is with Siligence and HELM as an enabler of unified entity registration system. So today's agenda, we're going to give a brief industry perspective, very brief, and then we will have a speaker introduction. You'll get to introduce Dr. Jimbo Lee, and then Siligence presentation, and then we'll have a Q&A. So rules of engagement, we're going to address all questions after the presentation. You can either put your questions in the Q&A box, or the chat box. If you have to leave early, we'll make sure that your answers get back to you. Or you could watch the webinar as it goes on demand. It'll be recorded. We will moderate the questions with the audience and the speakers, and then everyone will have access, like I just mentioned, to an on-demand recorded webinar afterwards. So our brief industry perspective from 2015 Visioneers. Just some background, just to kind of ease everybody into Dr. Lee's discussion today. So small molecules, building blocks, scaffolds, preclinical, clinical candidates, metabolites. These are a low molecular weight, less than 1,000 Daltons, organic compounds that do something in a biological process, or the hopes is that they will do something. And then large molecules need new ways of handling them. So we're talking DNA, RNA, protein, antibody-drug conjugates, oligos. You just go down the line, vectors, plasmids, cell lines. You know, the list is actually quite large. And then you have, like I mentioned, the antibody-drug conjugates, which are a small molecule and a large molecule together. And some of the newer or new modalities that companies are inventing, again, are a combination of a small molecule and a large molecule. That list is growing. You could think of even vaccine as a multi-component entity, where you have different things in your vaccine that are used to generate a response. So entity registration is started many years ago, maybe about 30 years ago, in an electronic format, with companies being able to do that. Cartridges were developed, the code was developed, the ability to store these molecules. It's been quite a while. However, don't underestimate the complexity around all of this. Chemistry is complex, and the biology and the large molecules add even another set of levels of complexity. And it is still a challenge today to be as complete as possible when registering properly molecules. Before the electronic environment, companies came up with very ingenious ways, paper-based ways of registering their molecules. There's a story of a European-based company where they almost had wooden dowels, and they had paper, pieces of paper with the molecule on it. And where you push the wooden dowels through, if it was an amine, you would pull back a list of pieces of paper that were all amines, right? So before electronic, there were very ingenious ways of keeping track of uniqueness in molecules from that perspective. Was it as complete as it is today? Probably not. So what is the purpose of entity registration? Chemistry, biologics, materials, you can go down the line. Well, the most important part of this is uniqueness. You want to uniquely identify a molecule. And the reason for that is, once it's uniquely identified, you can use it in processes or discovery and know exactly what you're dealing with. And it's critical because there are so many molecules that are almost the same molecule, but not quite. And so that's the reason for the uniqueness. You can now electronically search accurately molecules in an exact search, a similarity search, a substructure search, etc. So now with the proper environment, you can search, which means you're going to have a fairness to your chemistry and large molecule environment. So you can find it, you can access it, you can interoperate with it, and then most importantly, you can reuse it. Pedigree, how did the molecule evolve? Knowing this is, again, a very important step when you're into the weeds and into the details of drug discovery. You really need to understand that pedigree, where things came from, how they were transformed, why they were transformed. The associated or ancillary cheminformatics data or bioinformatics informatics data, again, critical pieces. When you're looking for that needle in the double haystack, you really have to understand the weeds and the details in order to be successful. Otherwise, just like in every other part of the organization, you would have to add more and more people to accomplish the same goals. But with an entity registration system, you can be extremely efficient, extremely accurate and precise, and it doesn't become a bottleneck in your drug discovery process. And then lastly, safety. You know exactly what it is. Maybe your MSDS sheets are associated with those molecules. You can quickly see them, but you can quickly identify the molecules you're working with, what their safety levels are, and it's understood. And that's a very important thing in drug discovery today. So with that, I'm going to introduce you to Dr. Jimbo Lee. He is the CEO of Siligence. He's had more than 20 years experience in pharmaceutical research. He was the head of chemistry at Ensemble Therapeutics. There he was responsible for molecule modeling, cheminformatics, medicinal and synthetic chemistry, process chemistry, and analytical chemistry. Prior to Ensemble, Dr. Lee led a multidiscipline team, including medicinal chemists, X-ray crystallographers, molecular modelers, pharmacologists, and DMPK scientists at Wyeth Pharmaceuticals. He's also worked at Boehringer Ingelheim in the areas of medicinal chemistry and combinatorial chemistry. So as you can imagine, Dr. Lee has a deep knowledge of the different therapeutic areas like oncology, metabolic diseases, neurodegenerative diseases, infectious diseases, inflammatory and autoimmune diseases, etc. He's an expert in research informatics and chemistry, and structure-based design and drug design. He received his PhD degree in organic chemistry from Princeton University. He was the year 1997 recipient of the Roche Award for Excellence in Organic Chemistry, and he has many publications in journals around the world and holds a number of patents, patent applications. Dr. Lee is the real deal. So he not only lived it, did it in practice, but then started his own company to help enhance the research in scientific informatics and has done an incredible job with their platform. So I'm going to hand it over to you, Jimbo.
Thank you, John. Thank you for the kind introductions. Let me share my screen. Can everybody see my PowerPoint slide deck?
Yes.
Okay. Yeah, as a scientist, as John mentions, you know, I feel the pain of a lot of colleagues here attending this seminar when we need to support, you know, various drug discovery program. And that's how I get into informatics, you know, about, 12 years ago, 13 years ago. And the meant to actually, you know, make life easier for all the scientists in my camp. And so today we are mainly focused on, as John mentioned, is really a unified solution to handle or support all modality in the current drug discovery and development environment. If you think about drug discovery, is actually dated back, you know, thousands of years. And the first actually therapeutics that used in patient, you know, could be as early as 2006 BC. And you know, as a form of a recipe of a mixture of different plant-based ingredients. And you know, and most of the 19th century, especially late 19th century and 20th century, a focus on small molecule drug discovery, as John mentioned, you know, drug substance that you know, have less than 1,000 molecular weight. You know, as you know, as the field actually understand, you know, more about atomic theory, molecular theory, and also biology, and that's the kind of the focus in 20th century, most of the 20th century. And so I think a lot of pharmaceutical company actually develop small molecule drug modality and then have been approved and used by patients. But how about nowadays? As John mentioned, starting 30 years ago, there's an electronic form of, I would say, drug substance tracking. And if you look at, nowadays, the drug modality or entities, as John put it, it's very diverse. And let's take a look at what AstraZeneca are working on nowadays. And this is actually a link to the AstraZeneca website. And you can see the modality class are very diverse. And under each modality, there's a class, there's different type of modality. So for example, PROTAC is not a typical small molecule drug modalities or entity. It's actually a conjugate of multi-component that represented by small molecule structures. And if you look at antibody therapeutics, certainly you have sequence-based antibody, but you also have conjugate-like type of antibody, for example, bispecific antibody, and certainly a biologics and small molecule hybrid type of multi-component entity or modality, such as antibody-drug conjugate. And if we look at the peptide and protein therapeutics, certainly complex peptide. One example could be the bicyclic peptide, sometimes it's tricyclic as well. And nuclear-based therapy, oligonucleotide conjugate, antisense DNA, mRNA, maybe most of us have the mRNA in our arm as a form of a COVID vaccine, and more. So as you can see, it's actually very, very diverse in terms of modality, drug modality that pharmaceutical companies are pursuing. And that's not only to the big pharma, but also to the biotech as well. If you look at Amgen's, one example of the large biotech, their entity, they're basically their drug discovery pipeline, and you can see very similar diversity in terms of drug entity or drug modality that biotech are pursuing as well. Now the question comes down to, with the evolving database system in the digital format, and how do you support all this type of diversity in terms of drug modality or drug entity? And before we take a look at that, the challenge is, let's take a look at the 2021 sales number in terms of the best-selling medicines or medications. And you can see in this particular chart here, the top 4 sellers is they are biologics, 2 vaccine and 2 antibody-based therapy. And if you look at the top 10 list here, only 3 small molecule drugs. So you can see the shift in terms of, I would say, the drug discovery pipeline to higher diversity and to more sophistication as well. Okay. And so with that background, really, how do you actually support such a diverse entity and modality in a drug environment? We mentioned some of those modality type here, and can be oligo-based, it can be peptide-based, can be sequence-based in terms of drug modality, and a lot of them is also involving in formulations. So, for example, lipid nanoparticle is one example of sophisticated formulation to deliver oligonucleotide-based therapy or vaccine into patients. So how do you support that? And what's the current situation in the R&D environment, especially in big pharma and in large biotech? Is what you will commonly see is separate system or application for different drug modality or entities. So when a new entity coming into the R&D environment, I think the IT and the informatics group will spin up a new application specific to that particular modality. It's standalone, and you may end up, a company may end up with dozens of systems to support, from the IT and informatics perspective. And all those systems may not talk to each other. Most of them don't. So you have data silos. So basically they cannot talk to each other. Now you end up with a very expensive environment. You have to support dozens of systems, and you have data silo that cannot be used across platform, across system, and you cannot use those data in downstream processes either. So this is really a headache for R&D, especially if a company needs to support such a diversity of entity in their drug discovery environment. And one example is, in typical big pharma or large biotech environment, I think they will have a system to specifically support small molecule. And that type of system are relatively mature through the development and evolutions in the last 30 years, and but still have some certain limitation. Many of the systems actually have trouble handling enhanced stereochemistry, and we can come back to that. What do I mean by enhanced stereochemistry? And then with the evolution of antibody-drug or recombinant protein therapies, so there is legacy systems that are purely sequence-based, based on natural sequence of those biologics, and that is mainly support sequence-based biologics. And there's no underlying that sequence. There's really no atom bond relationship of each of those, I would say, biomonomer, such as amino acids and nucleotide, and they are purely sequence-based. And such a system, typically, this term are two examples of legacy system, typically don't talk to each other. They have, first of all, the underlying informatics supporting those two different classes of modality. There's nothing common. So they cannot, first of all, they don't need to talk to each other. And second is that they cannot talk to each other. And now with the evolution of more sophisticated modalities such as mRNA, siRNA, antibody-drug conjugate, how can you support such such a sophisticated modality? As I mentioned here, this is really one of the common pain points that we are facing in a drug discovery environment. Okay. And just use my Pomasin as example. This is Ionis antisense DNA drug molecule. Is actually a oligonucleotide. Okay. If you want to actually adapt the legacy system, in this case could be the small molecule registration system to support such modality, that's what you see in terms of the Pomasin structures. It's really making chemists dizzy, and biologists will turn away. They don't want to see structure at all. So I think it's really not I would say productive to kind of retrofit some of the legacy system to support highly sophisticated macromolecule as drug modality. Okay, and now what can be a potential solutions? And here are some consideration when you look for a solution that can support diverse modality. And a few pointer here is one has to be intuitive to scientists. Right? I think coming from a drug discovery background, especially in the area of medicinal chemistry, has to be intuitive to chemists. But chemists is not the only class of scientists in a drug R&D. Also have to be intuitive to biologists as well. And downstream, right? I think data scientists, it's really important that it's friendly to data scientists as well, so that the information that captured in a informatic platform is machine readable and learnable, so that you can actually use data for downstream processes and the so-called fairness that John mentioned. Okay, and now how do you bridge that gap of the small molecule world, which is purely atom bond based with support of stereochemistry, versus a purely sequence-based world of biologics such as antibody and recombinant protein? The answer to this, the good news is that in the last 10, 15 years, there is a new standard coming into play. And John mentioned that briefly in his intro. It's called the hierarchical editing language for macromolecule. Okay, and that will be one of the potential solutions for bridging the gap. Okay, and what do I mean by hierarchical editing language for macromolecule? I think the key word here is the hierarchy. Okay, if you think about a macromolecule, one example here I show on this particular slide is a conjugate of oligonucleotide with peptide bridging by a chemical linker. Okay, so the top of the hierarchy in this particular example, we call it complex polymer, because this is really have multi-component, multi-category of component in such a biopolymer. Okay, you can call it biopolymer, you can call it chem polymer, it doesn't matter to me. So that's the top of the hierarchy. Okay. Now the second layer of the hierarchy, what is this complex biopolymer composed of? It's simple polymer. So for example, simple polymer could be this oligos as a component in this complex polymer. Okay. Now you go further down the hierarchy, what is actually make up of this simple polymer? It's nucleotides. Okay, and the nucleotides will be the monomer in this hierarchy. Okay, and if you look at nucleotide, it can be broken into 3 subcomponents as well: the base, the ribose, and the phosphate linker. Okay, and the representation of those monomer is basically atom bond relationship with stereochemistry support, a typical small molecule work. And if you go further down the hierarchy, it will be atoms. What compose those monomer in terms of atoms? Okay, so now you can probably envision this is actually could be a bridge, right? I think the monomer represent in small molecule representation, a biologics representing sequence-based representation, and the HELM technology actually bridging that gap of structure-based and sequence-based modality. Okay, on the right-hand side here, you can see that this is actually the HELM string to represent this complex polymer. And if you look at the nucleotide, this is the HELM string for the nucleotide component. Okay, so this, I think, in my view, this is actually a good option to bridging the gap that we talk about. Okay. And you may notice in John's presentation is actually this HELM technology was discovered and developed by our Pfizer colleague, and the lead author is Tianheng Zhang. Yeah. Okay. All right, now let's come back. How do we represent mipomycin in a HELM representation? So this is the structural representation of mipomycin, and this is the HELM string representation. This is the machine readable format of mipomycin. Now it's much more simplified. Okay, you can identify the component easily. I think even biologists may like this. Okay, look at the sequence-based likeness of the HELM structures. But underlying each of the monomer or monomer component, chemists actually can drill down to the atom bond connectivity using the HELM notation. Okay, so this is great. I think we may have find a solution to bridging the gap, and but how to implement that, right? I think this has been international standard now. A lot of big pharma, large biotech, and government agency and publisher has adopted this notation to represent sophisticated macromolecule, and here's the ISO number for that standard. Okay. Now, how do you actually implement such a solutions? There's a couple of challenges actually, right? I think because it really needs some in-depth technical know-how to do the implementations. And also there's a tool-based barrier too. So one of the example is the user interface. Historically, when HELM was developed at Pfizer, and a Java-based thick client was used to support such a technology, and that is really make it difficult to deployment and support. Okay. Another area is that in order to utilize the HELM technology, each company or each organization will need to establish a well-curated monomer libraries. That is your basis to build your polymer, basically, either simple polymer or complex polymer. And I think you also need an intuitive platform to manage the monomer library too. I think John mentioned about uniqueness. Not only the uniqueness of the entity is important, but the uniqueness of the monomer in your monomer library is also very important to make this technology implementation work. Okay, and third area is really your organization will need to have the IT resources if you want to implement HELM by yourself. Okay, not only have the IT resources, but have to have the informatic know-how to do that. So how we can make this type of implementation much easier? Can we develop out-of-the-box solutions, so that you can actually do, plug and play, and maybe with some, simple configuration, depends on your specific R&D requirement. And the answer is yes. I think especially to address the first two pain points in terms of HELM implementation. Cytogen has been involved with Pfizer very early on, before it's actually Pfizer open sourced the HELM technology to Pistoia Alliance. And also we are the developer of the HELM web editor. So the HELM web editor was designed in a way that we closely collaborate with our scientific community in terms of the design of the HELM web editor. It's a JavaScript-based editor, HTML5, and it's targeting to meet I would say the comfort level of chemists and biologists. So on the left here, you can see, you have the monomer library, management on the left here, and then here is the HELM structure representation. And then if you're a chemist, if you want to drill down to the atom bond connectivity, then you can highlight a portion of the molecule or the whole molecule to look at the atom bond connectivity. So this is a solution to make deployment of HELM technology much easier. And the second challenge that I listed in the previous slide is really how do you actually manage your monomer library and especially share your monomer library with your collaborator outside of your organization. So we actually developed this helmmonomer.org. It's a .org nonprofit platform accessible to all the scientific community. And it serves two purposes. One is to give you a platform to actually get your hand into a starting set of monomer library, which is provided by Pistoia Alliance consortium. And second is to give you a secure environment to share your monomer library with your external partners or organizations. And the third area, this is actually served as a translator of a HELM string. So in order to understand a HELM string or even a HELM structure, you need to have access to the monomer library used to create a HELM string or HELM structures. Okay, so this is basically, as long as you have access to that monomer library to create a HELM string or HELM structure, you will be able to fully understand what that particular structure means. Okay. So that is kind of addressing the two pain points that I talked about. Okay. Now we have the technology to bridge the gap. How do you actually build solution out of it? I think, from Cytogen's perspective is that, we develop the underlying engine first. So the software development toolkits. So that is have three components. One is the front end, is our JavaScript sketcher, which is the foundation for the HELM web editor. And the back end is our MoSQL database cartridge that not only supports small molecule and sequence-based biologics, but also using HELM to support the macromolecule, the sophisticated macromolecule. And in the middle tier is our Mo engine .NET informatic toolkits. So with this new engine, then we are able to put together solutions for specific need in the R&D environment. And today we are going to mainly focus on the RegMol application, which is a entity registration system to support all modalities. But it's also, in addition to support modality or entity registration, this is also an assay registration system as well. So basically, you register your assay protocol, and now you not only have a database of all the modality or entities, but you also have your assay data captured and related to the modality being tested using those protocols as well. Okay, so let's take some example. Take a look at some example. What type of modality can be supported by RegMol, which is an entity registration system. Here's a few examples of small molecule and with enhanced stereochemistry support. So let's say that you have an enantiomer you want to register and create a unique ID. Now we come to uniqueness. How do you represent an enantiomer with ambiguity? So you can actually use the all one flag to represent that this is an enantiomer of two possible enantiomer, and this is actually a mixture of two enantiomer, and this is actually is one of the two diastereomer, and you have the trans relationship, and but you really don't know the SOR at the chiral center. And this is actually representing one of the four possibilities of diastereomer. Okay, so here are a few examples of the small molecule representation in RegMol. Now, how about peptide? Okay, you're getting a little bit more sophisticated. Here's the example of linear peptide with unnatural amino acids, and here is a cyclic peptide with a single cycle. And here's an example of a hybrid between peptide and small molecule conjugations. And you can think about, we call this inline HELM. So you represent peptide component using the HELM technology, and you're using small molecule representation for some of the small molecule component. And here's an example of the bicyclic peptide, and here's a tricyclic peptide. So using this framework, you are able to actually support very diverse modality. Okay, and here are some example of oligos. Can be a single chain oligo with unnatural nucleotide components, and you can be bicyclic, actually duplex of oligo, and with unnatural nucleotide. And here's an example of the hybrid, and we call it the inline HELM. So you represent oligo in the HELM format, but you can also have small molecule component in your representation as well. Okay. Now come to ADC. I think as John highlighted, right? I think this is I would say, multi-component entity that have biologic components, but also have small molecule component, for example, in the form of payload. And such a modality will require a system that can bridge the two worlds, or basically bridging the sequence-based world and the structure-based world. And in the middle is an example of, it's so-called the inline HELM. You have a peptide linker, with some chemical component with it as well. Okay, and this is not just a picture. I think it's really important to understand everything underneath is the machine readable portion of the informatic. So behind the antibody symbol is actually capture the sequence of that antibody and also capture the information about the attachment point or linker to antibody as well. Okay, and that come to, in addition to the underlying technology, we also develop a technology called conjugate builder to specifically support multi-component modalities. And those can be example of antibody-drug conjugate, peptide-drug conjugate, oligo-drug conjugate, PROTAC. PROTAC is a chemical component-based multi-component modality. It can be multi-specific antibody. So those are the modality that the component are purely biologics. But how do you actually, using John's term, how do you track the pedigree of all the component, the relationship of the component with the conjugate? I think that is actually supported by our conjugate builder technology. So what is a conjugate builder? It's come with the regimen. So conjugate builder is basically allow you to pull any component in your entity registration system together, just like you reach out to your toolbox. Depends on what type of project you are doing, you use different tool. And so in Regimol, you will have all the component for your conjugate registered, regardless what type of component that is. So on the left here, just show you a few examples of those components. And then using conjugate builder, you can bring those components into the conjugate builder and connect the component into a sophisticated conjugate. The example of antibody-drug conjugate or antibody-peptide conjugate and more. So it's really component agnostic. And this is actually, through that process, you can actually not only tracking the conjugate you are registered, but you also track the pedigree. You actually establish that lineage using the conjugate builder. And here's an example of ADC. Okay, and you can see that easily identify the registration ID. Now come to uniqueness, right? All your components are unique, okay, have a unique ID. And then when you put them together, it also track the genealogy, not only at the parent level, and this is a conjugate of an antibody with a linker payload. And the linker payload was put together as a small molecule drug conjugate. So you can drill down further which one is the linker, you can see here, and which one is the payload. Okay, yeah, and you can trace back the pedigree, of the conjugate. But that pedigree is not only applied to conjugate. I think this technology applied to all the I would say genealogy or lineage of any modality registered in the Regimol application as well. So for example, you want to see really which cell line actually generate that particular antibody and which vector actually used to transfect that cell line. So that lineage or pedigree relationship will be tracked in the Regimol application as well. All right. So I will end my presentation here, and happy to answer any questions you may have.
Fantastic. Thanks, Jimbo. Oh, I'm sure there's going to be a lot of questions. Let me go to the first one here so far. What is the current state of parser availability for HELM?
Excellent questions. So it's basically one of the challenges for HELM implementation is how do you convert some of the legacy representations of biopolymer, such as peptide or oligonucleotide, that containing a natural component into the HELM format. And that is really company specific, because before HELM notation become a standard, each company will have their own legacy format, how to represent those complex polymer. And so I think I call it, I think the question is about parser, and it's actually a translation layer. So our team actually have developed a variety of parser, and to do the translation depends on the custom format of the legacy syntax. And so that actually can be easily to support data migrations. So you don't have to sketch the HELM structures manually for each of the, say, the complex biopolymer. So those two are available, but will be specific to your specific format, basically.
Okay, great. The next question is around duplicate checking with HELM notation. Is that something that's done, Jimbo? Is that a process you could kind of describe maybe?
Absolutely. The uniqueness check, actually for HELM notations, is really critical, actually, to develop that technology. So you think about, if you look at biologics, right? If you look at biologic registration, uniqueness is typically sequence-based. Okay, if you have the identical sequence, that will identify that biologics is unique. Okay? But for macromolecule, that is a little bit more tricky. Okay, because that has a natural component in it. So if you represent, if you're just simply using the HELM string, let me move back a few slides. As a uniqueness determinations, you could actually create duplicate. If you're just saying that I'm going to using the HELM string, if the identical HELM string, it will be used as a uniqueness check, only the HELM string as a uniqueness check. You can create duplicate. The reason for that is that one simple example is the sequence, the order. If you flip A to B and B to A, okay, if you're just using HELM string, that will create two registration number for you, because sometimes HELM string doesn't actually keep track of the order of that particular sequence. So to develop uniqueness check, what Cytogen does is actually drill it down to atom bond connectivity. So we actually develop the algorithm to create a hash code to check the uniqueness. So you kind of drill down to the atom bond connectivity, just like small molecule. Yeah. Hopefully that answers your questions. Yeah.
Sorry, I was on mute. Yes, I think that answers the question. Does HELM handle a combination as a distinct entity, say a combination of antibodies?
Yes. So I think that come to it's mixture, basically, how to handle mixture. And so basically, that's I think what I would recommend is using the conjugate builder is to make that connectivity. You don't have to have covalent bond to determine uniqueness. So basically, a combination of antibody component or fragment, we can develop a uniqueness check criteria, a business rule to determine, the uniqueness of such combination as well.
Okay. So I have a question around integration. So, as you know, Jimbo, owning your own software company, it's very difficult sometimes because everybody in the industry is at their own point of evolution. And what you find sometimes is it's not always easy to kind of intercalate yourself into their environment. So how do you integrate with legacy systems or drawing packages or whatever it may be? Like maybe you could describe that a little bit for the audience.
Yeah, absolutely. So I think certainly, especially in a big pharma environment, and then some of them have dozens of systems to support different entities. It's not practical actually to, I would say, bulldoze down to the house and expect a new house will be built tomorrow. So I think our experience is that it's really introduce a system that can really address the current pain point first, right? I think in this case, it will be the regimen to support, for example, peptide, oligo, and conjugate. And then in the process is actually integrate regimen with your legacy system, for example, with your small molecule, registration system. so that you don't have to recreate those components for example, small molecule component to be used in the conjugate builder. Through integration, you can pull those components into conjugate builder. And another example is integration with the sequence-based biologics registration system currently in the legacy system. And then you can actually bring in the antibody component, into building a conjugate. So I think that it will be, our recommendation is not bulldoze down to your house. It's actually, build attachment first. And then later on, if you say, well, I really, tired of have the house that have so many attachments, then you can think about redesign the house and gradually ease into building a new house. So that's our recommendations. Yeah.
No, that actually makes a lot of sense.
Yeah.
There's a question here. Are there any open source Python, R, or Julia packages you'd recommend for working with HELM data?
Yeah, so if you look at the Pistoia Alliance website, you can actually have a software starter kit to implement HELM technology. So if you want to do your own custom software development, you can. And what is different here is that Cytogen actually is a developer of quite a number of those software components, and we actually give you a better starting point. So out of the box, more or less out of the box solution for you. And so for that, you really have to think about whether your return on investment, okay, whether you want to develop yourself, and invest in a lot of IT resources and software development, or you can actually start with a system, and then you can just configure it, and that may impact not only your costs but also your, I would say, development cycle or deployment cycle as well. Yeah.
That's good advice. There's a follow-up on the HELM interpreters. Is everybody developing their own, or is there a common code base?
Yeah, so the interpretation of HELM string is has a common business logic to it, and but each company actually have their own monomer library. Just give you an example. Let's say that I call alanine with the letter A, but company B call it B. So now you have two different letter or symbol to represent the same amino acids. That make the interpretation very difficult. So that's why I think we'll require a translation layer, or another alternative, not alternative, it's actually a good starting point is that if company can leverage the work that performed by Pistoia Alliance to put together, it's actually involving quite a number of large pharma company in that effort, and Pistoia Alliance actually released a HELM core monomer set, and that work is continuing, and that has been transferred to IUPAC now, continue to build that starting set of monomer, not only peptide, oligonucleotide, chemical monomer, and can be oligosaccharide, building block as well. So basically now, if we speak the same language, it make collaboration very much easier, especially across organizations. Yeah.
That kind of leads into a question I've been having, or wanting to know. So there's there's always pros and cons to standards, right? Yeah. HELM has survived, and it's actually evolved, right? And you just described some of that. The Pistoia picking it up, your involvement with that, it's actually taken it to a different level. In your opinion, and I'm only asking this because there's always backstories to standards and who lost out, who won. There were other things, at the time, but I think it's important that, you kind of adopt one and evolve it and use it, and try to get everyone to use it. That's the goal of a standard. So my long-winded question is, what are some of the pros and cons
Mhm.
to Helm, and
Yeah.
what are some of the things that are being done to eliminate the cons? Is that
Mhm.
is that a fair question?
It is a very fair questions. I think I can share why we actually, from Cytogen's perspective, why we actually adopt Helm in our
Hmm.
system, right? I think the reason for that is actually Helm is a architecture that is actually highly flexible, can be evolved, okay, to support a variety of, I would say, monomers. And with that, then you can actually support a large variety of, biopolymer. And, I think it started with, support for peptide and oligos, right? And if you think about it, the architecture of Helm can be used to support chemical polymer as well. So especially, chemical polymer that have, different, uh, component, I would say, right? And that can be used, and also, saccharide, polysaccharide as well. So those are kind of the framework that I think is flexible enough for it to evolve. Actually, it's not restricted to, what I mentioned before, is that, well, each time a new modality come in, you have to develop a new application, a new standard to support that. And Helm has the ability, in terms of its design and architecture, to be flexible, and and also support ambiguity as well. So that's my 2 cents. Why I think Helm win, in this standard competitions so far. Yeah.
Right. But I mean, the thing is that you and your company have probably worked with it,
Yeah.
applied it,
Yes.
as much, if not more than, maybe a private or a public organization, pharma company, et cetera, that may have taken it and done everything with it. So
Yeah.
you have some lessons learned.
Mhm.
Is there room for improvement, like say from a complexity perspective or a usability perspective, et cetera? Have you noticed things like that where you've been able to give feedback and say,
Mhm.
this is overly complex, or this isn't complex enough, or we
Mhm.
could make this into a more usable format? Just any thoughts around that?
Absolutely, absolutely. I think that, our experience is really collective wisdoms. Through our experience for implementation of such system at big pharma, large biotech, and small biotech, they don't do too much customizations, so they just take our solution and run with it.
Yeah.
Well, but for big pharma and large biotech, they have very specific use cases and challenges and pain point,
Mhm.
and a lot of them, they are common, very common pain point. Okay,
Yes.
so
Agreed.
so it's really a collective wisdoms, and such a collective wisdom actually drive the evolution of Helm implementations. And I think there's certainly a lot to learn, in the future as well. But I think those pre-competitive type of knowledge, you know,
Yeah.
can be shared, right? It really, really is. We don't have to reinvent the wheel. Each company reinvent the wheel, and then later on they cannot communicate with each other if they somehow form, collaborations. So I think that's what I'm looking at. Another thing is that, based on our experience, right? I think the standard, starting with the basics, but the advanced functionality has to be developed and
Hmm.
not necessarily has been included in the standard. So one example is the example of inline Helm, which doesn't come with, come with the standard kits.
Right.
How do you actually represent the different representation in a single pictorial display, and
Yeah.
underline Helm string? How do you track that? So those are the, I would say, advanced functionality is custom built. Yeah.
Got it.
Yeah.
And then if we can sneak in two more questions. So one is around the conjugate builder, which I find very interesting.
Yeah.
Almost like I can see this evolving into like a new modality builder, right? And then and packing that full of artificial intelligence and things that really help scientists build better conjugates, or just describe, tell us what you're doing there and what you're thinking.
Exactly. You know, I think you hit the snake right on the head, basically, John. Your questions, and
Yeah.
you know, it's not just how to, you know, you give an example of using you know, mechanical board to do uniqueness check, right? And the conjugate builder is actually the key thing is that pedigree that you talk about. You know, one of the key components is the pedigree, and so you can downstream for data scientists, they can actually do data analysis to discover which component actually contribute to favorable property of that conjugate, because those relationship
Right.
has been captured at the point of registrations. So using conjugate builder. Yeah.
Very cool.
Yeah.
How about years ago when I was doing more conjugate work and things like that, and the informatics around it, the science wasn't strong enough to actually identify attachment points, maybe, you know, on the antibody or etc. But we could kind of come up with ranges. Has the science improved where we're able to find definitive attachment points, or is it still at the point of ranges, and there's still a level of detail needed to be worked out there?
That's an excellent question, John. I think you know, for supporting connectivity, you know, we have to consider site-specific conjugations, but also have to consider ambiguity in the
Right.
conjugation side. So that's why make it, you know, challenging, right, for each company to develop the uniqueness check business logic. If you do it by yourself, I think it could be a headache for you.
Yeah.
So now I think
Agreed.
so if you utilize the collective wisdom, you know, from a lot of big pharma, large biotech, it may be much easier actually for such an implementations.
Yeah. So I don't want to miss this last question. How are Helm strings stored in the database? Are they blobs? Are they is it text? What is it?
That's a good question for our IT colleague. I believe it's actually stored as a Helm string in a database table, but I can get back to you.
Okay.
Yeah, and I'm a scientist, so I don't
Yeah.
write any code. So.
You're more than a scientist. Come on, don't you're very humble. Well, here's the deal. We're going to wrap this up. So people you know what, scientists underestimate the complexity in science. Obviously, the general public then really doesn't understand the complexity, but scientists need to realize that entity registration is still not 100%, and it's very important to get this right, because it is a foundational building block in your organization. The more detail you have in there, the better things are captured. You know, it's kind of like a downstream process that happens. If you have a hole in your boat, it's just going to fill up with water, and too much water at some point. And unfortunately, this is where a lot of companies end up when they don't do things right. And so this is a perfect example. Silogens has put forward here a solution that will help keep your data fair, will help properly register, and allow you to add all the details you want, all the business rules you need to add to be super successful. And just take it from my 30 years of experience, Jimbo's 20 plus years of experience. This is a critical part to any evolving R&D organization. Jimbo, you want to say anything?
Thank you, John, for the insightful comments. I think I really enjoyed the conversation at this webinar, and happy to address any questions that our audience didn't have a chance to ask offline. Yeah,
sounds great. All right, thanks everybody for your time today, and we look forward to getting this up and sharing it and putting it up as on demand. Take care. Enjoy your summer. Talk soon. Bye.
bye.